Good morning, everybody. Here in the Class of 62 auditorium and on Zoom. Welcome to Angle Rounds for May. This will be our final Angle Rounds presentation of this academic year. And it's my great pleasure today to be introducing Paulo Lizano as our speaker. But before I say anything about him, I want to give the usual housekeeping reminders. So just especially for the people on Zoom, your cameras and microphones are off. If you try to speak, no one will hear you. Please don't use the chat box on Zoom. That's forbidden. That's really for the panelists here. But we do hope that people have questions or comments at the end. And if you do, put them in the Q&A box at the end, and I'll read off as many as I can, depending on how much time we have at the end. Also, it's very important for people, everybody, whether on Zoom or here, to do the evaluation at the end. And if you're on Zoom, you'll see that a link to the evaluation on a slide come up at the very end of the presentation, and in that chat area. There'll be a link there as well. Okay, so now getting back to our speaker, who is Paulo Lizano, MD, PhD. Dr. Lizano is an assistant professor of psychiatry at Harvard Medical School. And in addition to that, he's a staff neuropsychiatrist at the Beth Israel Deaconess Medical Center, an affiliate faculty member at the Harvard Stem Cell Institute, the division chief for translational neuroscience at Beth Israel, and the co-chair of the Diversity, Equity, and Inclusion Committee, also at Beth Israel. Dr. Lizano received his MD and PhD in cell biology and molecular medicine from Rutgers University. He then completed his residency in psychiatry at the Harvard Longwood Psychiatry Program, and after that did a fellowship in neuropsychiatry at Beth Israel Deaconess Medical Center, which he finished in 2019. In the 4 years since then, he's been quite busy. He started out as a senior instructor in psychiatry. He's now an assistant professor. He received a K Award, a K23, to study brain and retinal impairments in early psychosis. More recently, received an R01 to develop a novel segmentation algorithm for studying choroid plexus morphology across psychiatric disorders. And he just found out yesterday that he's receiving another grant from the Burroughs Wellcome Fund Career Awards for Medical Scientists program to develop in vivo and ex vivo models for studying blood-brain barrier disruptions in early psychosis. In addition to that, Paulo also receives funding from the American Philosophical Society DeLand Fellowship and from the Milken Institute to conduct research using non-invasive brain stimulation to personalize treatments for patients with psychosis and bipolar disorder. Also, I wanted to mention he was the recent recipient of the Robbins Guzet Award from the American Psychopathological Association. In addition to his research interest and work, Dr. Lizano's worked with first episode psychosis and high risk for psychosis patients, has an interest in that type of clinical work, and has strong interest in brain stimulation techniques. Where, for those of you who aren't aware, we're hoping that he joins the faculty of our department, and so I hope everyone gives him a warm welcome, and I'm looking forward to a good discussion at the end of his talk today.
Thank you so much, Steve, for that introduction. And thank you, everyone who I've met with here and who have helped during our visit. I didn't come alone. My wife was also with me and my 2 kids. The city is beautiful. The resources here are wonderful, and the work that everybody is doing here is great as well, both from a clinical and a research perspective. And so I want to thank you all again. As was mentioned, I sort of live in 3 big areas of research. I'm not going to be talking too much about our blood-brain barrier research imaging, or blood-brain barrier imaging work that I'm doing. Mostly going to be focusing on vision and our transcranial stimulation work that has stemmed from that. And so the title of my talk today is Towards a Precision Psychiatry Approach to Targeting Visual Dysfunction in Psychosis. Let's see here in this slide. just the title here as well. Here are some of my titles that we went over already. And here are some learning objectives. And so we're going to learn about visual pathway dysfunction and psychosis, which may not be new news to some of the folks here in the room. And we're going to learn a little bit about inflammation and visual dysfunction. I realized I ran out of time to talk about that, so we're just going to briefly touch upon it here and its significance. And then we're going to spend a good amount of time talking about the treatment personalization using non-invasive brain stimulation. I don't have any financial relationships or conflicts to disclose. Happy to change that, though, if anybody in the room happens to have, money that they would like to offer. So, yes, and so this is the accreditation and certification statement as well. And here we go. If people want to check out our website, it hasn't been updated in the last couple of months, but feel free to peruse. You'll find our clinic there and our research program there as well. Created the Lizano Lab. And I want to take this time as well to wish a happy Asian American and Pacific Islander Heritage Month. I myself am of Andean descent from Ecuador, and so really appreciate it and appreciate the sort of the diversity here and how much people appreciate this kind of celebration for this month as well. And so we already went over the learning objectives, and we're going to jump right in, okay? And so I'm going to, hopefully there's enough time to go through everything that I put on the slides for this presentation today, and I hope people are able to learn about, the interesting work that's happening in vision science and psychosis and the translational neuroscience aspects of it. I think the brain is really cool. I think that there's some amazing things that we can do with the brain, and I think that our patients are in need of the kinds of work that we're doing, the kinds of intervention that we're doing. And this is sort of the next phase for psychiatry. So I'm really excited about it. I just went to a conference in San Diego where all they talked about was brain stimulation. And so this is something that it's great for people to learn about, great for people to get excited about. And, hopefully this is something that we can bring here as well to sort of continue to enhance the work that's being done. So I'd like to start off with a case for our clinicians in the room, including myself. And so case-based presentations are a nice way of getting people engaged. And so here, you direct an early psychosis clinic. I don't know who that is, maybe the person speaking to you right now. And we were asked to see a 21-year-old Hispanic male. He has a history of depression, anxiety, and first episode psychosis. He presents to the emergency room with a 3-month history of hearing multiple voices, thoughts about being followed by a white car and receiving messages from the CIA, and they were also noted to have some erratic behavior. They report that their vision is blurred, their objects are changing shapes, photographs seem to be torn into pieces, and images remain in their vision for a while, right? And so, this is a really, the patient has a lot of insight if they're able to tell you of these things, right? And so really, they're not going to report these kinds of things. And really the goal for the first half of this presentation is just work through what are some of the other visual symptoms that people might present and get in the habit of asking those questions as part of your assessments. Like many of you probably were taught, although maybe it's different here, that visual hallucinations are this complex thing that people see that they're really scared of, and it doesn't have to be just that. And we can start identifying these things earlier and start intervening earlier. And so how often do you see patients with psychosis who report related vision, right? So that's an important question. Is this even clinically important? And how often do you ask about other visual symptoms outside of do you see something that other people don't see, right? Which is standard, resident question, attending question when they're seeing patients. And in what ways could visual symptoms affect functioning, right? And so if you work with this population, you know that it's something that's tremendously impactful in their ability to, for them to carry out their daily living. And so often, when you hear about visual impairments, you hear about hallucinations and illusions. So I was alluding to this already, right? So hallucination is a visual percept that's experienced when people are awake. They're not elicited by an external stimulus, and it just kind of, comes to mind for an individual. And then you have illusions, right? That you're typically taught there's something in the environment that induces this stimulation. You have this misperception. There's this filtering apparatus deficit that happens that then leads to, experiencing things that otherwise may not be experienced. And so people have looked at this for a long time, right, in a variety of neurological disorders, ophthalmological disorders, psychiatric disorders, and in substance use. And so what the general teaching has been, right, is that, you know, in eye diseases and in visual pathway dysfunctions, you primarily get these like illusions or visual perceptual aberrations that patients might experience. And when you look at people with psychosis, they predominantly are, you know, you're taught that they have auditory hallucinations or that they have delusions and that they don't have these other sort of perceptual abnormalities that exist. And I'm here to hopefully convince you that that's not always the case, and that a lot of these patients, if you start asking, you're gonna find that they're having these visual misperceptions. As a side caveat, this also happens for auditory processing. We're not talking about that here today, but so something to be aware of. And so when we look at visual hallucinations, right, or visual system impairment, we can see in the general population about 7.3% of people report having some sort of visual perceptual abnormality, a visual hallucination. Some of this is cultural. Some of this is just, you know, if you don't sleep for a day, this is something that can be more likely to happen. And then you have affective psychosis like bipolar disorder, for example. And if you ask people, you know, do people with bipolar disorder have visual hallucinations? So, you know, overwhelmingly people are going to say, no, they do not. And that's not always the case. So the current estimates are about 15%. We've seen a bit higher in our studies where, you know, people with bipolar disorder have or report visual hallucinations. And then obviously in schizophrenia, it was estimated to be 27%, but that number seems to be actually higher than that. And so as I mentioned earlier, visual hallucinations are mostly described as being complex, negative in content, and they're interpreted to have personal relevance to the individual. And a lot of this is still, you know, relatively accurate for a lot of patients. You know, the figure's usually humanoid, and they make the person feel overwhelmed and scared, which is what generally brings them to your care. And I guess I'll say that, you know, visual hallucinations have largely been ignored. So a lot of people and a lot of research and effort has gone into understanding auditory hallucinations and auditory processing. So this is an exciting area, and it's exciting to be around people who study vision so deeply. In a large cohort of patients with psychosis spectrum disorder, so this is schizophrenia, schizoaffective, and bipolar disorder, we looked at their lifetime report of having either hallucinations or delusions, and we can see here that they have multimodal hallucinations. So people who have experienced visual hallucinations in the past have multimodal visual hallucinations, so we're talking about auditory, tactile, olfactory hallucinations. And then these people who've experienced visual hallucinations also have polymorphic delusions as well. They're more likely to have other types of delusions. So this is another reason why it's important to look at or ask patients about their visual symptoms. When you look at cross-sectionally what their symptomatic profile looks like, these patients who have visual hallucinations in the past tend to be more symptomatic. And so they have more depression, they have more manic symptoms, they have more positive symptoms and general symptoms of psychosis. And so overall, they also tend to have a higher risk of suicide and substance use as well. So this is an important population that we want to be carefully taking care of. This is some of the work done by folks in the group, so Brian Keene and Dr. Silverstein as well, where they looked at, you know, what I was talking about before, not just visual hallucinations, but are there visual perceptual aberrations that are happening? And how often is that happening? Is it something that's clinically important? And so utilizing a scale where you can assess these things sort of more specifically, they were able to demonstrate that in patients with schizophrenia compared to those who, you know, don't have either differentiated schizophrenia or they ended up being diagnosed with another psychiatric disorder, that they ended up having more visual perceptual aberrations that were reported. And this was reported in regards to, you know, the changing in colors in people's vision, in changing how they perceive other people's face or body, for example, and whether they noticed any movement in an object that was stationary. And so these are some of the things that you would hopefully be able to ask about. They also noted that, you know, the greater number of visual perceptual abnormalities you had, the higher the rate of hallucinations and delusions as well, which makes sense, right? Just based off of what we were talking about today. But this is important work demonstrating that, like, we need to be doing more. We need to be assessing, you know, visual perceptual aberrations in this way. And so when we're thinking about vision, right, vision is a pretty complex system in the brain and in the eye. And, it has this bottom-up processing. And so you receive a signal or some sensory information into the eye, and that gets processed in a way that allows a signal to get sent to the brain that can interpret that information to let you know sort of what the object is, or whether it's moving or not, or what color it is, or where it's oriented in space and time. And so there are issues that can happen at this bottom-up processing, right? When you're thinking about visual acuity, a lot of us, are wearing glasses or corrective lenses in the room. And so there's some sort of abnormality that's happening at the level of the eye or the lens that it's making it difficult for you to see. Same thing goes for being able to determine contrast or being able to read things that have lower contrast. Patients with schizophrenia have a harder time doing this. And same thing with determining how fast something is moving. And so these are things that sort of point towards evidence of having a bottom-up processing effect. But there's also top-down effects. When you think about, that information that gets processed through the eye, it gets sent to the brain, that has to get processed to let you know, how close is an object to you? Is it moving towards you? Is it moving away from you? How to make an image out of, sort of a complex signaling environment, such as here, where you're, able to determine whether or not there's a shape that forms based off of this sort of complex image. And so patients with psychotic disorders have a much harder time doing these kinds of things. And so a lot of time has been spent studying the top-down processes that occur in patients with psychotic disorders. And so we have a pretty good sense of, what part of the brain is doing what, and, how it might be impaired. And less is known about the bottom-up approach. And so this is a lot of the work that's being done by people here. So Dr. Silverstein, Dr. Keene, and others as well. And so, I've said a lot. Hopefully, this information is being received well. But I also want to, bring this back to how this might be clinically important, or how you might think about your patients that you're seeing that are expressing visual hallucinations. And, what does a medical or ophthalmological examination look like for visual hallucinations? So obviously, you're trained to get a really good history, a really good mental status exam. I feel like as a psychiatrist, we're great at that, and even as a psychologist as well, for those in the room, and nursing staff. But asking sort of more specifically about other visual symptoms would be important as well. We don't always refer to our ophthalmology colleagues, but maybe we should, or maybe there should be a line of care for patients not with just psychotic disorders, but with other psychiatric disorders or neuropsychiatric disorders where vision is a problem. Same thing with hearing. So if you're thinking about an elderly patient who is starting to lose their hearing or starting to lose their vision, they may be more anxious or more depressed, and they can't relate to you that they're having those problems unless you're assessing them. And we should be doing the same thing with our younger population, our early course psychosis population, for example. And so measuring things like visual acuity, color vision, visual field testing, looking at the back of the eye. The back of the eye can tell us so much about what's happening in the body. It's not just telling us about the eye itself. It tells you about, whether somebody has cardiovascular effects, what their risk of stroke is, what their risk of cognitive decline is. And so we should really be, taking a deeper dive and looking at what's going on at this window of health in the body. Other tests might include getting some blood work, getting some toxicology screens, and maybe even a CSF analysis, depending on what you're thinking this might be, right? If you're thinking this is potentially substance-induced, you're going to get a urine drug screen to rule out certain drugs that might be causing or resulting in these visual hallucinations. If you may do CSF analysis if somebody has an autoimmune encephalitis sort of picture where they're reporting these kinds of visual hallucinations and other neurocognitive symptoms. And then lastly, depending on if you're thinking this might be migraines or seizures, or they might have been a stroke, then you're thinking about, I need to get an EEG, right? I need to see what their electrical activity is like. I need to see if there's any focal lesion that's in the brain that might sort of indicate whether there's an organic cause to what's going on. And if mostly ophthalmologists will do this, but there's certain genetic disorders that are more likely to have visual issues, and you would get genetic testing in those cases. And so this isn't going to always apply to psychiatry. We're keeping an open mind about, how you're assessing this and whether or not it's something that is potentially problematic or may lead you to a different diagnosis, and making that correct diagnosis and treatment is going to be really important. And so here's a sort of a flow chart on some things to consider, right? I'm not going to go through all of it, but this is found in Up to Date. It's a really nice, I think, summary of what things to think about when somebody's reporting visual hallucinations. And so whether those are simple or complex, whether it involves one eye or both eyes, and are there other neurological or other psychiatric symptoms that might be involved? And so we're going to go to the next part of this. And so you see this patient, you start treating them, you notice that they're starting to get better. You ruled out some other basic things. You obtained a urine tox screen, you did some basic labs, or you collected some basic labs. Cognitive exam, they were on bedside cognitive exam, they were performing relatively well. And so your level of concern is much less, but you're like, okay, there's this study going on. There's this physician or this researcher who's conducting this study. I think you might be a good fit for that. And so you talk to the patient, they agree to participate in the research. And this research involves collecting visual biomarkers in psychotic disorders, primarily early psychosis. And as part of that, you undergo clinical, cognitive, and neuroimaging assessments. And so here's some of that. Here in this study, this is my K23 study that we're currently undergoing. We collect electrophysiology of the eye to tell us, how the eye is responding to signals that are being sent to it, what the structure of the eye looks like, and what the vasculature looks like, and I'll go into that a little bit more. But also, how does that relate to how the brain is functioning? So is the eye potentially a window into the brain, and how do we determine that? And so we need to be able to collect all this information to sort of build that kind of information out, at least in this population. Just gonna take a sip here. And so here's the bottom-up approach. So we're starting off looking at the eye, and the eye is made up, there's a lot of homology or similarity in structure between the eye and the brain. It's not one-to-one, it's not exactly the same, but there are a lot of similar cell types that are found in both, and they function in a very similar fashion. And receiving information, relaying that information forward, or receiving that back as well. And so this is an opportunity to be able to study the health of the brain and the body by looking through the eye. And again, I don't think we'll have enough time to go into all the details here, but this is where the eye is. You have a lens, information or light passes through there, it gets centered. Fovea isn't necessarily there, it gets centered. There's a blood flow that passes through there, and astrocytes, you get signals that get passed into the lateral genucleate of the thalamus, which is a major relay center, and then that information gets passed to the visual cortex, which we'll talk about. But right now we're going to be focusing on the eye itself. And so how did I get interested in the eye? It's actually really funny. My training is in vascular biology, as was mentioned. I was studying ischemic reperfusion injury and how do we prevent that. And then I made the transition to psychiatry, which was honestly the best decision I made. I love the population that I work with. The science is fascinating, and the people that treat this population are amazing. But I thought, what's the coolest thing you can do in neuroscience, right? And it was brain imaging. And I was like, all right, I want to learn brain imaging. I need to get my hands on some brain MRIs, and I want to look at it. And then I sat down, and this is what I saw. And it was kind of disappointing, right? It was just like, okay, there's all these, here it's portrayed on a two-dimensional image, but it's 3 dimensions, right? And so you get these voxels that are there, and it, depending on how many there is, it tells you about how thick or thin the cortex is, and that's about the information that you get. There's more fancy stuff that we can do, but this is how I started off, and I was like, I don't know if this is necessarily what I want to continue doing, right? This is, it's not enough information. How are we going to understand how the brain works? It's so complex. There's so many layers. There's so many neurons, astrocytes, microglia, vasculature. This is not going to give us the level of information that I would like to have in order to understand psychiatric disorders. And then I didn't come across this exact picture. There was a picture by Madeline Meyer in American Journal of Psychiatry that showed a fundus image of the eye, and I was fascinated. I was captured, from that point. But this was another really cool image that I found. I was like, okay, you can see this is sort of the inner part of the eye, right? And you can start seeing the different layers, and you can see the different cell types that are there. And there was just much more detail. It looked much more elegant. And I was like, okay, this is really cool. This is something that might actually based off of what I knew, from a neurobiological standpoint about psychosis, I was like, oh, I can actually like see different things utilizing retinal imaging. And those different things were this, right? This is sort of a depiction of what the eye looks like, right? So this is where light would come down this way, hit the back of the eye. This is where you have photoreceptors. Information gets processed and sent through these bipolar cells to the inner parts of the eye. And then that information gets relayed, back into the thalamus. And I was like, that's really cool. In psychosis, you have problems with synapses. Like, we can look at synapses in some of these layers. That's fantastic. I don't have a vascular image here, but we can see vasculature with some really high resolution, and that stuff was great. And so I was, obviously, I was smitten by, what possibilities existed by doing retinal imaging. And I was, really taken back by a lot of the work that Dr. Silverstein was doing, continues to do in this area. And so this was our pilot study. I took some patients from this large schizophrenia, or the schizophrenia spectrum study that was going on. Made a collaboration with people at Mass Eye and Ear, and I was like, I really want to scan people, and I want to scan their eyes. Can we do it? And they were really fantastic about it, and they allowed us to do that in about 50 people. And so we started off by looking at the macula, which you can see here, or fovea, sort of the general area that it's called. And so here we were able to image the different layers of the eye. And here I'm only reporting on 2 layers. These are the significant layers that we found. Here is the right eye and here's the left eye. So pretend like somebody's looking at you, right? And this is the outer nuclear layer where the photoreceptors are located. And then this is the outer plexiform layer, which is the synaptic layer we were talking about. And we started noticing that there were deficits, and there were regional deficits that were identified. And so in the ONL layer, there were reductions, and those reductions were pretty high, an effect size of 0.8, 0.6. That's really high. Most brain imaging studies, the effect size is 0.3, so not really that great. And so this was telling us about maybe there's a photoreceptor problem, maybe there's a synaptic issue that's going on, maybe vasculature has something to do with here. We also noticed that the ONL was correlated with cognition, and so the thinner the ONL was, the greater the cognitive deficits, and so that made sense and fit with what is sort of what we were hoping from a biomarker to inform us about not just symptoms, but cognition. And then what was really interesting about ONL, it's again, there's, it's a photoreceptor layer, and in particular, there's cones and rods that help process the information from vision. And so here's some work by Demon and Dr. Silverstein as well that demonstrated that there was one part of that electrophysiological response in the eye that was reduced in patients with schizophrenia, and that part coincided with cone response, and so this is part of the photoreceptor response. The other thing that was really interesting is that there's a syndrome called Usher syndrome, which has sensory neural hearing loss and retinitis pigmentosa. And when you look at that population, up to 23% of people can have a psychotic disorder. And I thought that was really, really interesting. And then I found this review that showed this, and I was just like, this is amazing. This is exactly what we find in schizophrenia. We find cortical thinning, we find cerebellar atrophy, ventricular enlargement, in the brain. And so, is Usher syndrome maybe potentially a good source to gain a better understanding of what's happening in patients with psychotic disorders? And then OPL, the outer plexiform layer, was really interesting as well, because it told us about oxidative stress pathways, which I'm interested in and don't have a lot of time to talk about here. Synaptic pruning, which is a major hypothesis in psychosis from the genome-wide association studies. And so this is potentially a hot area. Maybe there's, what we're capturing is high synaptic pruning that's going on, and this could be a potential marker. There are pharmaceutical companies trying to develop drugs to target synaptic pruning, to try and dampen some of that process down and hopefully be able to help patients. And then I'll say all of this with a caveat. So the other thing we did, since we scanned people in 2 different scanners, we were able to compare how well scanners did when you were looking at them or comparing them to each other. And we find that the major layer that gets studied, which is this retinal nerve fiber layer, that there was high variability. So it may not be necessarily the greatest place to start, but that some of the deeper layers were actually much better. to do that. And then we also found that the overall retinal layer thickness in the peripapillary, so this is this optic disc space, was correlated with overall cognition and functioning as well. So the thinner the layer, the worse cognitive performance and the worse functioning you'll have, which is really interesting. We performed an updated meta-analysis as well. So here's looking at the peripapillary layer. So I know I just talked about just ONL and OPL, but there's several other layers that are impacted as well. And we're still trying to figure out what the exact signal is, or where sort of the signal of high interest. And then when we look at the macula also has a variety of different layers that are impaired, with some of those standing out, like this GCL volume being a really high effect size difference in a schizophrenia population, for example. And then we'll move to talking about vasculature. So as I mentioned, I'm a vascular biologist by training. I think the blood vessels are really interesting and understudied in psychiatric disorders. Depression actually probably does a great job of this, where they study what the potential impacts are of abnormal vascular function, but less is known in psychosis. And so here you can see the nerve bundle fiber that's going to the eye. You have vasculature that enters, pierces through, and then enters to a variety of the layers of the eye. Here's a more sort of zoomed in image of that. So you have this choroidal layer, which is outside of the eye. On the outermost side of the eye, you have this choriocapillaris layer, which actually provides a lot of the energy, oxygen tension that's needed for this photoreceptor layer, which we were talking about earlier. That's really important. And then there's this deep capillary plexus and the superficial capillary plexus. So what was really nice is with OCTA, or optical coherence tomography, angiography imaging, is that you're able to image all these layers, which is, again, really cool. You can't do that in the brain. The brain, you sort of get, it's either high or low for perfusion. And this one, you're getting, down to the potential level of deficit, or where there might be microvascular dysfunction. And so we examined that in this same population that I was talking about before with psychosis spectrum. When we looked at it as an overall group, you can see there's some areas where there's reduction in perfusion, and then there's some areas where there's an increase in perfusion. And then when we split the group up into early psychosis, or chronic schizophrenia, we found that actually in early psychosis, there tends to be quite, a significant increase, sort of across the board almost, for vascular perfusion, which fit with sort of my hypothesis with inflammation and microvascular dysfunction, that early course psychosis is an area where you really want to try and target, where the neurobiology is very different than it would be if you were studying someone with chronic psychosis, where that inflammation, that microvascular dysfunction has already calmed down, that event that was leading to those symptoms have passed, and then all you're seeing after that is what's left over. So you see all this neuronal degeneration that's happening, and you get decreased vascular function. So others have demonstrated in chronic psychotic disorder populations that primarily there's a decrease in vascular metrics. So I think this is something that's really interesting. We found that these indices, so the greater the vascular indice, the greater the number of symptoms, so total positive psychosis symptoms as well as negative symptoms. We also looked at the choroid layer, so that was that outermost layer as well. That's really related to another sort of important area of research of mine, which is looking at the choroid plexus that I'll talk about in the next slide. But we found that there were sex-specific changes there as well, that there were alterations in females when you're looking at choroidal thickness, and then when you were also looking at vessel density and vascular volume. So this is really interesting work. As I mentioned before, I study this little structure in the brain called the choroid plexus. It's found in the lateral ventricles, the third ventricle, and in the fourth ventricle. It's been there a long time. It helps us. It's one of the main and first entry pathways into the brain. So when you're thinking about infection, immune processes, autoimmune issues that happen, toxins, it has to try to break through this barrier first, and then the blood-brain barrier. And so that choroidal finding that we were talking about in the previous slide may be related to this. I don't have a lot of time to talk about this, but we've identified choroid plexus abnormalities in people with clinical high risk for psychosis as well as chronic psychosis. And we have a sort of whole area of research building up on this end as well. And so coming back, so we talked about the eye, right? The bottom-up approach. Now let's talk about the top-down approach, which is something that we know a little bit more about, at least in relation to the eye and psychosis. And so you can see here, right, sensory information is sent to the eye. That information is relayed to the lateral geniculate nucleus of the thalamus, where most of the projections get sent to V1. V1 processes simple information such as contrast, orientation, and direction. That information can get passed on to V2, 3, 4, and V5MT. V5MT is something that is of high interest to us because it's involved with motion processing, but also we'll talk about some of the other findings that we've had related to V5. And so here in this large psychosis spectrum population, we were interested in looking at, well, what are the specific visual cortical abnormalities that are identified? People prior to this have done this in smaller sample sizes, and there was maybe signals that it might be increased or decreased. And so because we had access to the largest sample to date, we were able to identify that there were surface area changes in V5MT in particular, but also V1 and V2. Surface area is important because that's what's set, that's something that's set from your development, right? And so you're sort of set with a certain amount of surface area. And that's something that doesn't change a lot over time, or at least not as much as cortical thickness does. The cortical thickness is something that has a greater impact by the environment. So stress, lack of sleep, substance use, those kinds of things will have a greater impact on your thickness than they would on your surface area. But we also found that there were differences or reductions in V5MT as well as V1 and V2. So that was really interesting. And then we found that in females and in people with early onset psychosis that the deficits were even greater. And so that pointed us to being in the right direction. Since that time, there's a new segmentation tool that was developed by Stanford that allowed you to get a higher resolution of imaging as well, and sort of looking at some of the other areas of the visual cortex. For some reason, they named them differently. Or so HOC 1, 2, 3, 4, it's the same thing as V1, V2, V3, V4. It's sort of, they're defined, the borders are defined differently, but we won't get into that. And then also they looked at the fusiform gyrus, and they found that you can identify subregions of the fusiform gyrus. Fusiform gyrus is primarily involved in object processing and face and word processing as well. So important area of the brain. I'm not going to talk too much about it here, although we have some findings related to that. This is hot off the press data that was sent to me, I think, during my trip here, so I thought I'd share it. So we were able to replicate these findings in a second wave of this psychosis spectrum disorder study. And so we found that, V1, V2, V3 area was lower in patients with psychosis compared to controls, that they had lower thickness and volume in all of these areas, including MT, which is of great interest to us. However, we were unable to replicate this sex by group interaction, so we weren't able to find that females had a greater deficit. We found that there were some clinical correlates as well, and that there were cognitive correlates. So it really makes us feel like we're on the right path to studying this. There have been very few studies that have tried to connect that have done retinal imaging longitudinally, so more than just one moment in time, and that have related that to the brain itself. So what's happening in the brain? Is that the changes that are happening in the eye the same as they're happening in the brain? Is one, is the eye having more sort of accelerated change than the brain? And so this was one study that did the first aspect. So they did repeated imaging of the eye 3 years apart. And this graph is a little weird, but it's really intriguing. So when you look at blue, these are their probands, or their patients with psychosis. And if you look at the blue markers here on the left, so this is the degree of change. So they had a 50 micrometer change over 3 years. Generally, the change that you see is in like the 0.4 range that they see here in healthy controls. So you have a significant increase in change. And so this is something that you can see by repeatedly measuring the total thickness of the eye. So this is something that's really impactful, something that you would not see something like this on the structural brain MRI scans that I was showing you earlier. When they looked at brain structure in that same study, they looked at the change over time in healthy controls. And so if we look at visual areas, which are here, right, there aren't a lot of changes that are reported. So in healthy people, the visual cortex, doesn't change quite a lot. And then when you look at patients with schizophrenia, you can see that there's this V5-MT reduction that's happening. There's this V1, and it's bilateral. There's this maybe fusiform area change, maybe some V1 effects that are happening. And so those are areas of significant change that are happening over the course of 3 years. Unfortunately, they did not look at the rate of change between retinal and brain, which would have been really nice to compare those. We've looked at in our data the ONL finding that we had, and so remember we had a reduction in ONL, the ONL was thinner, and we found that that correlated with the whole brain matter volume and with white matter volume in particular. And so at a cross-sectional level, we can say, these two things are related. Our study, unfortunately, wasn't cross-sectional, so we couldn't look at the rate of change. We also looked at perfusion, and so how does perfusion in the brain relate to perfusion in the eye? And so this is a cool image that I was talking about earlier. But you only get this sort of cross-sectional image. It doesn't tell you anything about the depth of the brain and sort of where the perfusion alteration is happening necessarily. But what we were able to do is we were able to demonstrate that the MT perfusion is altered in patients with schizophrenia. They seem to have higher levels of perfusion. We don't know what that means, but it's something that we're currently interrogating. We also found that there were some fusiform gyrus issues in face and word processing areas. And then we looked at relationships. So sorry, some of it got cut off. So we looked at, the overall density of vessels in the eye and how it related to perfusion in different lobes of the brain. And it turns out the frontal lobe ended up being the one that was most significantly related. And that was really interesting in proband. So it seems that this increased perfusion that we were seeing is related to increased perfusion that's also happening in the brain. So maybe there's this sort of sequestration of blood flow that's happening, or maybe there's some other process that's happening at the frontal lobes that may have other impacts on patients with schizophrenia. And then we sort of deduced that a little bit more, found that the superficial layer of the eye also had this effect, as well as the deep, but we didn't see this in the choriocapillaris layer. So this is really interesting because when you look at the perfusion in the brain perfusion, at least for a part of it, comes through this inferior carotid arteries. There's this ophthalmic artery that gets sent out, and this is what can perfuse at least a large proportion of the brain. You have this middle cerebral artery, and then you have these anterior communicating arteries, which provide blood flow to the frontal lobes of the brain for the most part. So it kind of makes sense, this relationship that we're seeing. We're looking to see, could this be a good biomarker for cognitive functioning or overall functioning in patients with psychosis? I thought these were really cool. So this is eye perfusion, brain perfusion. There's a lot of similarity that exists between those two. And as I mentioned before, we have a longitudinal study where we're doing ERG, structural, vascular imaging. We're doing brain imaging as well. And so we're really excited to see what those results look like. So I guess this is just to say that the eye is potentially a good proxy for the brain and can tell us what's going on, and may potentially be a therapeutic, has some therapeutic potential in the future. All right, so hopefully we're doing good on time. I think it might be a little short, so we might go through a little bit faster with the second half. So this is treatment personalization for visual dysfunction. And so in this case, you're asked, later in the day in the clinic, you're asked to see this 31-year-old patient who has, treatment-resistant symptoms. They're continuing to hear tanks on the streets and officers stalking them in the subway. ECT was indicated, but the family refused, and he had a legal guardian. And so what pharmacological option or treatments might you consider in a patient population like this, right? And what are some non-pharmacological things that you might recommend? And, if you were gonna do non-invasive brain stimulation, where are you gonna target, right? That's a really important thing in brain stimulation research. It's like, where are you gonna target, with what, when, how, sort of all of the questions which makes it a really intriguing area. And so if you're thinking about pharmacological options, it really depends on what the underlying cause is. So if somebody has delirium tremens from alcohol withdrawal, you're going to treat them with benzodiazepines. That's going to help resolve some of that. If the patient has dementia, you might use agents such as memantine or cholinesterase inhibitors, or even antipsychotics to help treat some of these symptoms. And so I won't go over all of these, but you have pharmacological options depending on what the cause is. However, in schizophrenia or psychotic psychotic disorders, we don't really have a lot of options. We treat them with antipsychotics and that's it. And not everybody gets better, or not all of their visual hallucination symptoms go away with antipsychotic treatment. And so, I talked about where to target, right? And so there's been this huge causality gap in psychiatric disorders. Everything that we've done and that I've showed you here has all been correlational, right? What does that tell you about like what's actually happening in the brain? And can we figure out, or is there a way to figure out how to make that causal link between some event happening and a symptom? And then can we utilize that information to better target the brain? This sort of goes over that causal, there's the causal inference, which we'll be talking about. There's this compensatory mechanism where you have some unknown process leading to visual hallucinations, and maybe that's associated with this change that's seeing in the visual in V5MT, this motion processing area of the brain. Or is it an epiphenomenon? So there's some unknown process and you're getting visual hallucinations. You're separately getting this, this brain activation change in this V5MT area, right? And so this is where we've lived, this compensatory epiphenomenon. I think there's some data to suggest that maybe there might be a causal link. And we can go back, right? So 100 years ago when people were looking at stroke lesions and trying to identify what part of the brain is associated with expressive language issues, for example, which was really interesting work done by Broca and Milner. And then there was a surgeon named Penfield who, during neurosurgical procedures was sort of stimulating different parts of the brain and seeing what happens. And he was able to detail out the homunculus, so which part of the brain, did what, which was really great work. And then brain imaging sort of came on the map, and so now you don't have to be as invasive, right? And then we have these non-invasive brain stimulation tools. So you can have this cause effect now. So you can, measure, perturb, measure. It's really cool, sort of what you're able to do, and you're able to tap into some of the dynamics of the brain by doing this. And so there's work done by Michael Fox over at Beth Israel Deaconess Medical Center, or at least he was there. He's now at Brigham and Women's Hospital. I trained with him in the Center for Non-Invasive Brain Stimulation. So what they did, they sort of took the typical neurological approach, right? Like, where is the lesion for psychiatric disorders? And so in the past, it used to, people used to think that the lesions had to be primarily in a sort of close by area, right, in order to cause a particular symptom. And it turned out that wasn't necessarily the case. You can have lesions happen in a variety of different areas. So you can have frontal, you can have this like parietal occipital area, temporal, and that can all lead to the same symptom profile. In this case, let's say visual hallucinations. And so because of efforts by the NIH to map the brain and to get, really great, densely, informative information about the brain, we're able to make these maps of functional connectivity for the average population. So you can see, like, what is the brain doing? How is it talking to itself? How is it talking to other parts of the brain? And then they were able to sort of reverse calculate. So they were able to map on these lesions that were causally linked to visual hallucinations, and then map it onto this human connectome functional connectivity map. And then, you get this bilateral extrastriate visual cortex region, V5MT, that turned out to be significantly causally linked with visual hallucinations. And so this fit nicely with our prior data. It fit nicely with some imaging studies where they put patients in the scanner while they were experiencing visual hallucinations. To no surprise, V5 lights up in addition to other visual regions. If you look at electrophysiology, looking at the brain dynamics and seeing what brain frequency is altered. So our brain, right, I wish I had more time to go into it, but there are different frequencies that do different things that communicate across the brain. And so in this study, they did a motion processing task, and they found that the delta range was associated with motion processing, which is, again, related to V5MT. So we were like, okay, this is really interesting. This is lining up really well. Non-invasive brain stimulation could be a way to sort of assess the function of V5MT. And so we used transcranial electrical stimulation. I'm a very practical person. I like things that are scalable, and so things like TMS are, kind of hard to get patients in to do that kind of work, especially if they have a psychotic disorder. Whereas transcranial electrical stimulation you can do in the comfort of a patient's home, and there's studies doing that in depression. But here, for those people who don't know, there's a transcranial direct current stimulation where you're able to, provide an anodal stimulation. So this would be stimulatory, or cathodal stimulation, which is inhibitory. You can do alternating current. So this looks more like how the brain frequency looks like. And you can do this at a particular brain oscillation as well. I'm not going to go into some of the other ones, but some of the other ones are also really interesting. And so the important thing to note is that this kind of stimulation doesn't induce an action potential, it just makes it more likely for an action potential to fire. There's been a lot of work demonstrating how this works for transcranial direct stimulation. So it has effects on neurotransmitter function, depending on whether you're doing anodal or cathodal. It can have changes on microvascular function, inflammatory processes, as well, astrocytic function, synaptic plasticity as well. It's really safe to do. There have been, thousands of sessions that have been performed over, X amount of time, and it's something that's really safe. The thing that most people report is maybe a little bit of burning and a headache at the location where the stimulation is happening. And so what people have done in the past, right, for brain stimulation, there's 2 case studies doing this, where they take the typical location, the left dorsolateral prefrontal cortex, which is what everybody uses to stimulate, and then they put the other electrode or sponge on the occipital cortex, and they look to see, are there any effects? And they did find that there was a reduction in auditory and visual hallucinations in these cases. And when you look at the current flow modeling for that, you can see like a large swath of brain is being stimulated. So do we really know what the specific effects are? And so this like old method of using sponges that you put on the brain to stimulate is really not necessarily a great way to better understand how the brain is functioning, how you might design a therapy for a patient's benefit. And so here in the corner, you can see it's not the exact device that we're using, but it's very similar. It's called high-density transcranial electrical stimulation. So you can see here, you can get higher focality of stimulation. So you can try to design it so that you can target that specific brain region that you're interested in. And we did just that. We took the coordinates that were provided to us in that paper that linked visual hallucinations causally to this extrastriate region. We used computer software to help us model what might be the best montage to use. And so we used cathodal in the bilateral extrastriate and anodal in frontal. Here's what the current flow model looks like. This is a lot better than what we saw before. Still not perfect. We have a device now that allows us to gain even greater focality than this. We did a crossover study. So some patients received cathodal inhibitory stimulation, and then they were crossed over after a month to receive delta stimulation. So delta is important for motion processing, as I mentioned earlier. And so what do we find? We found that in a small, this is pilot work, but we found in a small pilot study that there was a rapid reduction of PANS general symptoms or PANS general psychosis symptoms at day 5, and that we found that with alternating current stimulation, you had a reduction at 1 month, which is really interesting. So maybe TDCS has some acute effects, while TACS might have some more longer-term effects. And so those things are really interesting. When we looked at self-reported scales, so for the clinicians in the room, we're terrible, right, at determining how well our patients are doing or not. And so using self-reported scales can be really important. And we've introduced that into our studies. And we find that at 30 days that TACS is able to reduce paranoid ideation and can reduce anger and hostility as well, which is really interesting to us. We'll skip this. One thing that's important to note here is that brain stimulation has been used in Charles-Bonnet syndrome using different montage than we did, but they found that they were able to reduce symptoms in patients with Charles-Bonnet syndrome who are having visual hallucinations as well, and that there might be a way to use the brain frequencies in the brain to determine who may be more likely to respond than not. So you can imagine that in psychosis, this is the kind of work that we want to be, or this is what we want to be working towards, right? Because we don't think that this is going to necessarily work for everyone. Now, being mindful of the time here, this is, I think, was really interesting. These patients also performed a steady state visual evoked potential where we flash lights in the eye and we look to see what the response is in the brain. We found that cathodal stimulation, to no surprise, reduced that response or the amplitude of that response, and that wasn't necessarily seen in the alternating current. And then we found that this change in voltage was also associated with the reduction in PANS general symptoms, which is really interesting. So you have this target of engagement, you have it being sort of linked to symptomatic reduction. So we think that's really important in the work that we're doing. We had this working memory difference as well that we identified, and so we found that with transcranial alternating current, that at 1 month, we were actually able to get people up to close to normal. Actually, this is compared, this is a Z-scored normative scale. So we were able to get somebody who's, people who are on average at -1 standard deviation up to close to healthy. So that's really interesting, and we're trying to investigate why that might be the case. Other studies have tried to do something similar. They used a different montage. They found there was no effect on working memory. And we just think maybe they're not targeting the right location with the right type of stimulation. So maybe delta stimulation is the way to go. We looked at some biophysical properties, didn't really find anything there. There's this whole body of literature that says you need to be engaging in a task or engaging in some sort of mental process during the stimulation process to enhance the effect. And though that's something that wasn't built into our study, but definitely future studies will include information like that. And then interestingly, we found that, so earlier I showed this finding that, actually it was a separate finding that Dr. Silverstein found. He found that there was reductions in this retinal electrophysiological effect at S3. And when you stimulate folks, they were actually able to improve and hopefully improve close to a healthy control population. So with the same stimulation montage. So we thought that was really interesting. Here's actually that work. All right, so hopefully I've convinced you that, causal lesion network mapping is a great way to potentially start targeting the brain. There's still a lot of work to do, how we're gonna stimulate, where we're gonna stimulate, with what, for how long, what can people tolerate, can we do it at home? There's so many open questions, which makes me really excited about this field. Obviously, this couldn't be done alone. I work with a lot of students back at my home lab, and I've had the support of the department and other junior faculty there as well, and obviously couldn't have been done without support, financial support, and then obviously couldn't do this without my family. So I'm glad that they were here. They were able to explore. This is Theo actually walking around outside. She likes her dandelions, likes to blow them away, and thank you so much. And hopefully there's some questions.
Thanks. All right, thanks, Paulo. We have a very short amount of time, but let's see. Brian, you have a question?
So this kind of touches upon a question that I brought up earlier this morning in our conversation. So you're part of the BSNP consortium, and so you have these different biotypes of psychosis. Biotype 1 has, increased negative symptoms, worse social functioning, gray matter thinning extending into occipital cortex. Would you expect increased retinal nerve fiber layer thinning in biotype 1?
Yes. So in biotype 1, they have identified that there was lower cortical activity. That lower cortical activity was associated with, or they sort of were able to replicate that there were brain changes associated with them, those being thinning. So I would imagine, yes. We didn't look at that actually, because our participants that we ran in our pilot retinal study would have that information. That's something we can go back and look, but we haven't looked at whether or not there was any specific biotype effect.
Thanks.
Bill?
So I was struck by the very bottom-up question that you brought up. The possibility that a major part of what's going on is at the level of photoreceptors. And the reason that I think about that is when, with all of these retinal degenerative disorders, you lose your photoreceptors, and one of the things that happens, people get a lot of hallucinations, and there's a huge increase in activity in the inner retina between the ganglion cells and the bipolar cells. Is that possible that that's going on here too? One of the reasons that I think about it is that there's now a very good method called the optoretinogram, where people can look at the function of individual photoreceptors. And so that gives you possibly a very early marker.
Yeah, I would leave it to the expert in the room. That's not my area of expertise yet.
Well, unfortunately, we're just about out of time. We're going to look into that. Seriously, we are out of time.
Yeah.
There were some good questions that came in over Zoom, and I will send those to you, Paulo, over
Sounds great. Happy to
email.
look at them.
And we will look into the optoretinogram. So thank you all for coming, and thank you everybody on Zoom who attended. Remember to fill out the evaluation. The QR code's on the screen there. And hope everyone has a good day.
Thank you.